to kind of grow, essentially. There's many subtypes. We're not going to look at the subtypes. I just want you to understand that if you look at CSFs, colony stimulating factors, what are they in charge of? White blood cells. Okay? I'm going to ask that question that I asked earlier because it depends. Well, I'll ask it now. The lymphoid white blood cell is part of what lineage? Did I give the answer right?
Yeah.
The lymphocyte. Sorry, delete that from your brains. The lymphocyte comes from what lineage? Okay, good. You guys got it. Good. Are we comfortable with these hormones and growth factors and what they do? Okay, perfect. Here's what we saw in lab, right? So if I showed you this on a quiz, you should be able to, for sure, tell me red blood cells, white blood cells, all that kind of stuff, yeah?
What does the CSF do again?
Oh, colony stimulating factor, it's going to regulate white blood cell production. Okay, yeah. So that should be pretty straightforward. Okay, let's kind of focus our attention now on the red blood cell, the erythrocyte, okay? So RBCs, their shape, remember they're biconcave. That's the way they describe them morphologically, right? They're biconcave. Do they have a nucleus? No, they lack a nucleus. Why do they lack a nucleus? Because they carry oxygen. They've sacrificed all their organelles to basically do one thing and not consume it, right? Don't get high on your own supply, right? Who said that? Scarface, right? So these guys don't get high on their own supply, right? Meaning they don't use up the oxygen, right? They only carry the oxygen, which is crazy. They don't use up any oxygen. They don't have any nucleus, nothing. That's why when you look at them under a microscope, you saw they just look like this red donut almost, right? Okay, that's the way they look. Because their job is to do what? Transport gases. That's their main job, right? Their number one job. And we've talked about hemoglobin, right? Hemoglobin is going to reversibly bind with oxygen. And we talked about the significance and importance of hemoglobin because that's an indicator of what? Iron, clinically. And I know the levels usually vary. Any lab, any book will tell you the average is going to vary a little bit, 12 to 17, 12 to 18, right? Depending. But I want you to understand that each hemoglobin molecule, so if you look at hemoglobin itself, right, is going to be comprised of an iron atom, an iron ion, I should say, which allows each molecule to bind to 4 oxygen molecules, which is pretty important. And we'll talk about that in a subsequent slide. You guys notice in this picture here, this is a capillary. And if you notice, these guys have a circumference of about 7.5 to 8 nanomicrons. These capillaries sometimes have a circumference of 6, right, or 5. So how does something that's a little bit bigger get into these little capillaries? What does the red blood cell have to do to get into this capillary? It folds in on itself, right, a little bit. It's kind of flexible and pliable. There's a protein, a special protein called spectrin on these red blood cells that allow it to kind of be really like floppy and fold in on itself so it can get into those capillaries pretty easily, okay? If we don't have spectrin, what can happen to that red blood cells that's trying to do this? Break. What's the fancy way of saying break? Lyse. So the red blood cell will lyse, okay? Lyse means to break down, to basically explode. So can you guys think of any diseases where the red blood cell is very fragile and lacks spectrum? I can think of 2, right? Thalassemia and sickle cell. Yeah, sickle cell disease or sickle cell anemia, right? Where the red blood cell is fragile, so it's trying to get into these small capillaries. What happens? It lacks spectrum. It basically lyses, okay? So spectrum is pretty important as well. So are you comfortable with this, the dynamic of a red blood cell so far, okay? Let's zoom in closer to this. So if we look at the biochemical, right, composition of a red blood cell, right? If we look at it under a microscope and we look at the biochemistry of it, we're going to see it's going to be comprised of 2 things. What is a red blood cell comprised of? 2 things. Basically hemoglobin, the heme and the globin, right? So what's hemoglobin or HB? That's the iron-containing oxygen transport metalloprotein that's found in my red blood cells, right? And each hemoglobin molecule has 2 parts to it. It has the heme that gives it the nice little pigment bonded coloration, the redness, to the globin chain, right? And at the center of this, at the center of the heme, is what? If you look here in blue, what do you find at the center? It's highlighted in orange for you guys. What is that? The very center. Iron. You guys see the Fe plus? Fe plus means iron. Okay, iron. So the heme central iron atom binds to how many oxygens? It's going to bind to 1, but then we multiply. You're right, 1 each. And how many globins or how many chains do I have? 4. I have 2 alphas, 1, 2. Do you guys see that? And I have 2 betas. You guys see that? Okay. You guys follow so far? So how many globin chains do I have? 4. Okay. And the central iron atom binds 1 oxygen to each of these, right? So each hemoglobin molecule that we're seeing here, this is 1 hemoglobin molecule. So each hemoglobin molecule can transport how many oxygens? 4. Very good. 4 total. And guess what? We have about 300 million hemoglobin molecules per red blood cell. So do the math. How much oxygen can you carry?
A lot.
A lot, right? A lot, okay? That's why for the most part, if everything's working right, you never even pay attention that you're breathing. You don't pay attention that anything is off, correct? Yes or no? I don't know. You guys are healthy, right? You guys are. Talk to somebody that is not healthy. Talk to somebody with emphysema or pulmonary congestion or congestive heart failure. Ask them how they feel, if they can breathe okay. You're gonna see a big difference, yeah? Okay, big difference. So again, it's important to understand the way this kind of works. So heme again, where do we find well, not heme, but if we look at the structure of hemoglobin, there's 2 parts to it, heme and globin, right? Heme gives it the pigment, because heme means what? Red or redness. And at the center of heme is what?
2 iron atoms.
An iron atom, okay? 1 iron atom that you see there, Fe plus, right? It's iron. And each central iron atom can hold how many hemoglobins? If we're talking about 4 chains here, 4, correct? So 4 irons equals how many oxygens? 4, right? Per hemoglobin molecule. And how many hemoglobin molecules do I have in each red blood cell? 300 million, right? So an insane amount of oxygen total, right? This is pretty intense biochem real quick, yeah? Side note, right? So wrap your head around this one, that's pretty crazy, okay? So basically the majority of the weight of the red blood cell, about 99% of the weight of the red blood cell is what? Is iron, basically. That's the weight, that gives it weight. What's the purpose of iron again? To carry oxygen. Clinically, what does that matter to me? Yeah, if they have low iron levels, what does that mean to me? They're going to be saturating low oxygen? Maybe, yeah, maybe. Maybe not, but for sure they're going to have what? Low iron, which equals what? Anemia. What type of anemia? Iron deficiency, which is the most common type of anemia that you're going to encounter, right? How do you treat? Red meat. What if I'm a vegetarian? I don't eat red meat. Greens. Leafy greens have a very high content of iron. Yes. Kale, spinach, there you go. There you go, Jules, you got it. Talking healthy. Broccoli? A lot of calcium, I think a lot of calcium in broccoli. But leafy greens, a lot of iron. Okay, I'm gonna skip over this. We're gonna see this in respiratory. The only thing I'm gonna ask you guys to just remember, if I ask you anything, is just terminology. So when oxygen unloads, it's called deoxyhemoglobin. Does that make sense? Because you're unloading oxygen, deoxyhemoglobin. When it picks up oxygen, it's called oxyhemoglobin. Does that make sense to you guys? Deoxy and oxy. Good? That's it. I'm not gonna ask you any of this stuff here. Just, we're gonna look at that respiratory. Okay, specifically now looking at erythropoiesis. What does erythropoiesis mean now? It's different than hematopoiesis. Hematopoiesis means all blood cells, yeah? Erythropoiesis means what? Specifically what? Red blood cells. Because again, look at the prefix. Erythro, or eryth means red. And then poiesis is formation of. So formation of the red blood cells. So what hormone, if you guys remember, is in charge of red blood cell production? EPO, erythropoietin. Very good. Excellent. Okay? Excellent. What lineage or what stem cell does the red blood cell arise from? Myeloid. Very good. Myeloid stem cells. Good. Quiz questions? Perhaps. Okay? So let's look at the process real quick. I'll kind of go through some detail with you guys, and I'll show you guys what's relevant for you guys to really know. So myeloid stem cells, again, there's different phases, the proerythroblast, but basically in about 15 days, that proerythroblast develops into this kind of like basophilic structure called an early erythroblast, and then a late erythroblast, and then it ejects its nucleus and becomes a normoblast. And at the very end of about 2 days, we have I should say at the end of 15 days, we have what's called the reticulocyte. So this whole process from stem cell to reticulocyte is 15 days, okay? And that reticulocyte is gonna mature into an erythrocyte in 48 hours, in 2 days, basically, right? So the thing I want you guys to remember here is this. What is the immature circulating cell from the red blood cell? What is it called? A reticulocyte. Okay. How do you know it's immature? There's still little. You guys see those little things there? There's still little organelles in it still, right? It hasn't matured fully. I think I have a picture of that I'll show you guys later. Oh, here it is. So do you guys see a blood smear? I don't know if you're able to see it with the light, but there's these little flecks, these little spots on there. Do you guys see those little spots on that reticulocyte? That means what? If you see this on a red blood cell, it's immature. What do we call that immature circulating red blood cell? A reticulocyte, okay? We commonly see an elevated number of these in anemia, right? Because your body's trying to push out red blood cells that are still immature to try to compensate for the lack of oxygen availability, essentially. How long does this process take from stem cell to reticulocyte? 15 days. And how many days does a reticulocyte take to mature to a full-blown erythrocyte? 2 days. Perfect. Perfect. Excellent. That's what I want you guys to know. Yeah, for the quiz. I do talk about it in the recordings about these stages, but for the quiz, those are the 2 things I want you to be aware of. Okay. Only 1, maybe 2% of all your circulating red blood cells are reticulocytes. You know, everything else is mature. Make sense? Good. Okay, so when it comes to the hormonal control of erythropoiesis, we got to know what hormone is in charge of all this. So erythropoiesis. So I just thought about this right now, right? Where are blood cells actually formed? Oh yeah, you guys know. Okay, good. What type of marrow? Red marrow, very good. Okay, and what stimulates those what stimulates the red bone marrow to produce red blood cells? EPO, erythropoietin. Okay, very good, excellent. You guys are good. Who makes EPO? Yeah, Talia's right. The kidneys make EPO. Very good. I like that. You guys are sharp. Goodness, you guys are too sharp. I don't like it. Making me nervous. You're making me nervous. You're too sharp. EPO. Very good. EPO comes from the kidneys. So where is EPO produced? Kidneys. Very important to know where these hormones are produced. We're going to talk a lot about hormones and their production. Do you think it's important to trace these things back? For sure. Quiz questions maybe? For sure. Know them. Okay? So very good. So essentially, when we become hypoxic, right? You guys remember what hypoxia means? Low oxygen levels. We're saturating low. We usually saturate like at 99%, right? Is it cool, Brooke, if we saturate like at 97% oxygen? 96%?
Borderline.
Yeah. Not too good, right? I mean, we want to see it at 99%, essentially. Anything that's kind of dipping down below that, we start to question as to what's going on here, right? When we become hypoxic, that's a big stimulator for the kidney to do what? Make what? EPO. Because it thinks what? What does the body think? There's not enough what? Red blood cells. I'm hypoxic. I need to make more. So let me make some more. That's the way it kind of works, right? So what type of feedback is this? Negative feedback. Okay, good. Okay. So what would cause hypoxia? What are some common things that you'd see that would cause hypoxia? Like, that you'd see in the clinic or hospital? Bleeding. Someone's bleeding and their hemoglobin levels are dripping on the floor, right? Because they're bleeding out, blood pressure's going down, all that stuff, red blood cells are on the floor. Are they gonna be hypoxic? Yeah, because they don't have enough oxygen to carry nutrients to where they need to get to. It's an acute setting. We gotta triage that, stat, but that's one cause. What else did we talk about that's pretty common? Iron deficiency, super common to see that. And then anytime there's reduced availability. So let's say we all take a trip up to the mountains, right? Colorado. We're all from San Diego, we're used to the weather. We're at sea level, right? We're chilling. That's different than if we go up to the higher altitudes of mountains, correct? What is less available in those areas?
Oxygen.
Oxygen. So we're all healthy, right? Hopefully, thankfully, okay? We're all healthy. We go up to the mountains. Are we gonna be in a hypoxic state? Yes or no?
Totally.
Yes. Yeah, because we're from here, right? And we go up there and we go for a walk. Can you keep up? No, you're gonna be out of breath. Or maybe I'm out of shape, I don't know. Maybe, because I tried doing that and I was out of breath, right? I was out. I can't. Why? Because I'm in a hypoxic state. I don't have enough what? There's not enough oxygen available for me to take in and do what I need to do, right? So they call this, basically hypoxia due to elevated altitude. But some common causes. Does that make sense? So all these are stimulators for what? EPO production, right? Excellent. Good. You guys are good. I just thought of something else. So if I had a patient, right across the street is a clinic that they deal with dialysis, right? And what does dialysis mean? That means what organ is shot? Kidneys. Kidneys aren't working anymore, okay? So if I had a patient who had kidney failure, right? What's one of the signs or symptoms I would find that patient in? What would they have? How would they present? What would they have? Anemia. Okay, but now we're gonna talk pathophysio. Jules, why would they have anemia? Because kidneys make EPO. And if the kidneys make EPO, and in this case they're not making EPO anymore, what can't they do? They can't stimulate what? Bone marrow to make red blood cells. Now red blood cells can't do what? Carry oxygen. So how's the patient present to you? With what? Anemia, right? They're gonna be hypoxic, they're gonna be anemic. How do I treat? If you give them oxygen, good. How do I treat the underlying cause though? EPO. You give synthetic EPO. Yes or no? Yeah. Did we just discuss pathophysio? Did we just discuss treatment options? Pharma? Right? That's next level questions we have to know. Yeah? Cool, right? How many diseases do you know now? How to treat? That's pretty hardcore if you could treat someone with kidney failure, right? Like signs and symptoms. That's pretty good. Okay? Okay, if we take a look at the slide here, we take a look at EPO. We're just talking about synthetic EPO. So what are the effects of EPO? Rapid maturation of red blood cells. Usually, basically, you increase those cells pretty quick. And we already talked about, what we call blood doping, right? Natural blood doping. So as I said before, I went up to the mountains. I couldn't take a walk. I was out of breath. I'm like dead. At about 2 weeks or so, right? I acclimated. I was able to kind of do my thing. I'm fine. I'm good to go. I come back down here, and guess what? Go to the gym, go work out, go run. What happens?
You're a monster.
Exactly. I like it. I'm a boss, right? I don't get tired. I don't fatigue. Why? I have way more oxygen reserves. What do my muscles need? Oxygen, right? My oxygen stores do not deplete anymore because I have such a high reserve because I acclimated, right? And when I came back down here, where there's plenty of oxygen, what happens? My cells are just full on saturated. There's no period of epoch or anything like that. I'm good to go, right? Do you think athletes do that for reals? Yeah, they train that way. That's the natural way to train. Could you kind of do a shortcut and just take the synthetic EPO and then get those stores up in cycle further and faster and further and faster than anybody ever? Yeah, okay, but if they catch you, what happens? You're done, right? Lance Armstrong, right? How many Tour de France? Like 7? 7 Tour de France's, #1 champion, he's the best, he's the greatest.
He was taking EPO.
He was taking EPO. Then what happened? Done. 0. Nothing. Done. Okay? So, at an athletic level, yeah, cheating, but clinically speaking, that you guys gonna do, fully okay to give your patients EPO, right? Especially if you've got like kidney failure or something like that. Does that make sense to you guys? The way this kind of works? Okay? Okay. So let's take a look at this balance here. Okay, so here we have homeostasis, my normal blood oxygen level, we're gonna say it's 99%, correct? Okay, so now I have low oxygen for whatever reason. Let's say I have kidney failure or well, I got some, whatever hypoxic state, whatever it is. And my O2 sat drops below that. What organ is gonna be stimulated to make EPO? Kidneys. Okay, so the kidneys are stimulated here. They make EPO. EPO has its effect, or its target cell is where? Bone marrow. Red bone marrow, like you said before. Within red bone marrow, I have those red blood cells that we talked about, the lineage from the myeloid stem cells, and we make what? Red blood cells. How long do those red blood cells take from
15 days.
Okay, good. Let me finish my sentence. And then how long do they take to mature? You guys are on it. I love it. I'm getting goosebumps by your answers. I swear to God. Good job. Look at that. That's hardcore. You guys, you give me goosebumps right there. That's good. I love it. Okay. So now I get more red blood cells. Now the red blood cell count goes up. My O2 sat goes where? Back up. Now my body's happy. I'm back at homeostasis. I'm good to go. Make sense? Simple, right? Chill. See, 202 is fun. How does this whole system work? What biochemical feedback mechanism? Negative feedback. Just like Jules said earlier, where do we get this stuff from? Iron, really important. Nutrients are really important. Amino acids, lipids, of course, carbs, everything really important. If we talk about iron specifically though, okay, we need it from the diet, right? We acquire it from the diet. From where? Leafy greens, red meat, liver. If all the organs in the body, I know, look at Brooke's face. Me neither. Liver doesn't taste good. I mean, you gotta prepare it a certain way, you gotta fry it a certain way, I guess you can make it taste good. But most people don't like liver, why? Why does it, what does it taste like? Like iron. It tastes like metal, so we don't want to eat things that taste like metal. That means what? It has a rich source of what? Iron. Yeah. Okay. Bone marrow is pretty big right now. Everybody's eating bone marrow. It's got a lot of nutrients. One of the main nutrients is iron. Leafy greens, really important to keep in mind there. You know, like we said, green beans, nuts, legumes, broccoli, fortified cereals, some fish, red meats. Like Andrew says, and he likes his meat. Red meat though, red meat. All these things are basically fortified, right? What else is important for blood cell production? 2 other vitamins that are really important is vitamin B12 and folic acid. Super, super important. Why? Because we need folate, folic acid, to basically synthesize DNA and repair DNA. That's the main function of folic acid. And B12. And this is especially important in rapidly dividing cells. So where in our timeline are our cells most rapidly dividing and growing? Before we're born. In utero.
People who are pregnant.
That's why pregnant individuals need to take the prenatals, because what's in the prenatals? Folic acid and, well, B12 is part of the mix. Why? To prevent something like neural tube defects. Oh, wait, hold on, I just thought of something. Do you guys remember the neural tube?
Yeah.
Okay, what does the neural tube give rise to? The brain and spinal cord. So what can happen if you don't have enough folic acid? You get something called spina bifida, right? Spina bifida. Oh, really? Oh, wow. So Andrew had spina bifida. Spina bifida is a bit where your central nervous system doesn't develop properly because of usually the lack of folic acid or folic acid. And basically the spinal cord kind of sticks out. Essentially, there's like a tuft of hair that's basically exposed. So it's a very precarious situation. It's amazing that you're able to walk. Yeah, that's amazing. Most individuals end up like with problems in gait, like walking, or they're in a wheelchair or crutches. So for sure, wow, amazing. That red meat. Good, gotta be strong. But that's exactly true. With folic acid, it's really important. No joke, folic acid is a requirement that all females, all individuals that are pregnant have to take to prevent what? Neural tube defect. So that's why it's really important when patients come in, and especially if they're childbearing age, they really get assessed and we figure out their plans. Are you planning on getting pregnant? Are you done? What's going on here? Because sometimes we don't see them until a couple months later when they're already a few months in or pregnant, or they don't think it's a big deal because, you know, a lot of people don't know how much can go wrong in a pregnancy. It's insane that we're all born kind of healthy, you know, for the most part. Make sense? This guy right here? So that's just the thing. Iron is important for the diet, you know, that's my point there. Oh, this one's crazy. Okay. This one is crazy. So this is hardcore. This one here is pretty intense. So let's focus on this one because there's gonna be for sure a quiz question on the picture and then one or two questions that are based on this whole dynamic here. So let's kind of like wake up for a second and let's focus on this one. So we're gonna see why red blood cells are red, why urine is yellow, and why poop is brown, okay? It all deals with blood. It's crazy. So red blood cells basically live in circulation, right, for how many days? About 120 days. Like one red blood cell, when it's kind of like born, if you will, lasts in circulation for 120 days, okay? After circulation, the dead cells are removed from circulation from an organ called the spleen, okay? And the liver, but you know, primarily the spleen. And within the spleen, we have a special cell called the macrophage. So this is a closed, zoomed-in version of a macrophage, this globular thing that you're seeing here. Where's the macrophage? In the spleen. So what's it doing? It's taking apart the old red blood cell and recycling components, right? The body's a great recycler, yeah? It wants to reuse and recycle as much as it can and doesn't want to throw away anything. So do you think it can use the heme? Sure. Do you think it can use the globin? For sure. That's the essence of hemoglobin, correct? So we can reuse this stuff. So let's kind of go through it together, okay? Let's look at the pink part first. I'm going to circle things and we're going to follow along. So here we have the destruction of these dead or dying blood cells after 120 days. Who's doing this? The macrophage. Where? In the spleen, right? And what's going to happen there is going to split the heme from the globin, correct? You guys see that? We're splitting the heme from the globin, yeah? Okay, and then number 3 takes us to the breakdown of the globins to amino acids. Can we reuse those amino acids? Yes, for what? Protein synthesis, right? Where do proteins come from? Amino acids, yes? Good with that? Okay, so we can also look at iron. Iron is Fe2. You guys see that, Fe2? Iron is removed from the heme, and then basically it's removed and then transported in the form of something called ferric iron. You guys see Fe3 here? What is that called? Ferric iron. And it binds to a transport plasma protein. So this plasma protein is found where? It's called transferrin. It's found where? In blood, correct? So basically my question is, how does iron get transported in blood? Does iron get transported as iron proper? Or does it change a little bit? It changes a little bit into something called what? Ferric acid, and it couples with transferrin. Transferrin transport, right? What's it transporting? Ferritin. Ferritin is what? Iron. Make sense? Okay? So far so good? Okay? And that's gonna bind, or not bind, but it's gonna go where? Where's that combo of ferric acid and transferrin gonna go? To the liver. Very good. It's gonna go to the liver, and here inside the liver, ferric iron is gonna detach. You guys see the detachment is gonna separate from transferrin and then latches on top of ferritin for storage. Because where is the majority of my iron stored? In the liver. So we're gonna use this as a basically backup, correct? So how do we store it? or where do we store it, I should say? In the liver, okay? And then basically, once we need it again, right, once we need it, it's gonna be released from its storage site from the liver. It's gonna go where? Into blood. Again, transported as what? Ferric acid and transferrin into circulation, into wherever it needs to go, into the GI tract, for example, okay? And then it's gonna go where? To the bone marrow, correct? Because where do I make blood? In the bone marrow. So it's gonna go to the bone marrow, okay? And then it's gonna basically undergo hemoglobin synthesis, and it's gonna use a mixture of, globins, of vitamin B12 that we just saw, folic acid, and of course the hormone that stimulates all this is what? EPO. Does that track with you guys, kind of? Does that make sense, kind of? If it's kind of a bit overwhelming, I can review it again, or you can listen to the recordings, because the recordings say the whole thing, and you can rewind it 1,000,000 times if you want. Make sense?
Yeah.
Okay, so do you think you should probably re-listen to this again, maybe?
Yes.
For sure, yeah. Don't space out on it. Pay attention, okay? Okay, let's go back here and let's look at the split here between heme and globin, and we're gonna focus on the heme now, okay? So when iron basically is removed from the heme, the non-iron part, because we just talked, all about iron, the non-iron part, you guys see this here? Is basically converted into something called biliverdin. You guys see this here? What color is biliverdin? Verde, green, okay? And bilirubin, it's gonna convert biliverdin into bilirubin. Bilirubin is like a yellow pigment. That bilirubin basically is gonna enter circulation, is gonna go to the liver, okay? And there, basically, it's gonna degrade this yellow pigment as bile. So from bilirubin, it converts it into bile. So what's one of the main jobs of the liver? What does it make in terms of digestive function? It makes bile. It converts the bilirubin into bile. And what's bile for? The breakdown of fat. So when I ate those pizzas yesterday and the nachos yesterday, what organ was working overtime to break down the fat? The liver, because what was it releasing? Bile, okay? So there's another little organ called the gallbladder that stores the bile, but who makes the bile? The liver. The gallbladder doesn't make anything, it stores. Make sense? So we're understanding where bile is coming from, correct? From bilirubin, which is a byproduct of the breakdown of hemoglobin. Yeah, so far so good? Okay, so the liver basically secretes that bile, right, into the small intestine when I ate those nachos and those pizzas. Plural, right, pizzas. And then what ends up happening? That bilirubin, because we've got bacteria in the gut, correct? It's gonna break it down from bilirubin to urobilinogen, and then sterobilin, and then basically feces. That change from urobilinogen to sterobilin basically is accomplished by bacteria, and that kind of converts that yellow pigment into a brown pigment, or dark brown or black pigment. And that's why your poop is brown, right, basically.
Oh, wow.
Yeah. And we also see urobilinogen excreted in the kidneys as urobilin, and that urobilin is basically a yellowish pigment, and that's why your pee is yellow, right? So it's basically a very scientific way to explain why your poop is brown and pee is yellow, right? It's all coming from what? The breakdown of red blood cells, right? The recycling of these elements of hemoglobin. So I would really recommend you guys re-listen to this again if you didn't capture everything.
So what happens when you have like blood in the urine or blood in the feces?
So if we're taking a look at like blood now in urine, it's going to change we haven't seen the urinary system, so when we get to urinary for sure, we're going to talk about all the pathologies there, right? But for now, let's just understand that usually urine is like a light straw color. It's almost the same color as your plasma, if you're well hydrated, right? If you're dehydrated, it takes on a darker amberish kind of color. If you're overhydrated, it's going to be clear, right? If we have blood in urine, it usually is going to look as kind of like a cola colored or tea colored, right? That usually means there's an issue that's going on usually above the ureters. So if this is the kidney here, you've got the ureters and you've got the bladder, right? There's an issue that's probably happening at the level of the kidney above the ureter. If it's frank or overt bleeding, that's usually due to trauma, right? So if it's really red, that usually means there's trauma. Something happened. You got hit in the groin and now you're peeing blood. That's common to see, right? Catheter placements, have you guys ever placed catheters? Right?
Yes.
If you take out catheters, sometimes clots and there's bleeding because what? There's trauma. So if you were to do a urinalysis, if the patient were to pee, it's going to be what? Kind of red, frank, overt redness. If it's dark colored or tea colored, that usually indicates maybe some type of infection. The most common thing that also comes to mind is like something like glomerulonephritis, like an infection of the glomerular capsule, the functional unit of the kidney, right? That's pretty common. Post-streptococcal glomerulonephritis is very, very common, especially in kids that are like post-strep throat 2 weeks out, and they start presenting with like tea-colored urine. You got to go back to the antecedents of the history of the infection, and then seeing if that's the root cause. So that's the potential cause of urine. If it's feces, again, depending on color, if it's frank, overt red, that usually means it's from everything from the stomach down, right? Because the stomach digests. So if it's black, that means it comes from everything from the stomach up, this here, okay? If it's black. Anything from the stomach down, that means it's not been digested yet, so it's gonna come out red. So we gotta find out what the bleeding is. Is it, hemorrhoids is the most common cause of bleeding, by the way, right? It could be something worse than that too. I'm giving you something acute. It could be cancer, right? It could be polyps. It could be many other things. If it's dark colored, that means that you might have a bleeding, like maybe an esophageal bleed. Maybe your esophagus is torn, meaning not esophagus, but blood vessels in your esophagus. Maybe they're bleeding into the esophagus, which goes to your stomach. You digest that blood, it turns black, and you poop it out. So it just depends on where the problem is. Yeah, black tarry stools, not normal. Anybody ever take Pepto-Bismol, right? And then look at your poop. What color is your poop? It's black, right? Because the bismuth salts in the Pepto-Bismol basically kind of messes with this process and converts your stool from brown to black, basically. So if you ever next time you take Pepto-Bismol, look at your poop, and then see how black it is, and then remember it and say, oh, okay, if my poop is ever this black when I'm not taking Pepto-Bismol, that means that I have some type of upper GI bleed. That's what it means. Good? Good.
Is this pictured in blood test?
Yes. 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 So yeah, review it for sure. Okay. Same information, just a different slide. This one's not on the quiz, but same information. All right, let's take a look at erythrocyte disorders. This is the good stuff here. So anemias. Anemias are characterized by a deficiency in our red blood cells that can originate from a couple different causes. What are the most common causes? Blood loss. We can have low red blood cell production or high RBC or red blood cell destruction, okay? So let's take a look at anemias, because anemias, there's many, many different types of anemias, correct? Right? So we have to figure out where they fall. Is it due to blood loss? Is it because we're not producing enough? Is it because we're destroying the red blood cells, okay? Do they all have some similarities though? Yes. Is there a decreased amount of oxygen carrying?